Hello, everybody. So my name is Nisha Naveen. I'm a sophomore at Wilcox High School in Santa Clara, California. And before I begin, I just want to say a shout out to my mentor, Ambika Verma, for all the help that she gave me for this project. Okay, so my project is called Battling Canine Parvovirus with an Antiviral Peptide Therapy Proposal. So I know it's a mouthful and you may not know anything about the topic, but I hope I can give you everything you need to know. So to start, I want to introduce the problem itself, which is canine parvovirus. So you may or may not have heard of it, but if I have any dog owners listening today, you guys may know what it is because when your dog was a puppy, it probably had to get a series of 4 shots to prevent their ability to get canine parvovirus. But if you still haven't heard of it, no worries. The first thing you need to know is that it's a gastrointestinal virus, so it affects the stomach and the intestine. It's transmitted through fecal matter. It can cause severe illness in infected individuals. It has a super high mortality rate, and most importantly, it still remains incurable, which is why finding an effective treatment method is critical to limit the evolution of the virus and also conserve the endangered species that it affects. And then on the other side of my project is the solution. Is the solution antiviral peptide therapy, also known as APT. So APT uses peptides or short amino acid chains, and these chains will then be designed to target a specific virus and prevent its replication. It's currently being used for COVID, herpes, influenza, and many other viruses that you guys may have heard of. However, why does that keep happening? Okay. However, it's never been used on animals or to address CPV2, which is why this research is very different. However, according to my research that I have been doing, it does prove to be more effective than the current therapies being introduced on the market for CPV2. And here's just a short amino acid chain or antiviral peptide. Okay, so the first thing that I want to address is the problem itself, which is canine parvovirus. So a really significant aspect of CPV is the fact that it has been evolving rapidly over time, which is why it's even harder to develop an actual cure for it. So the first strain, known as CPV1, was developed and evolved from feline panleukopenia virus. The significant part of this is that it only affected young domestic puppies around the age of 3 weeks old. However, over time, it developed into CPV2, and today CPV2 affects wild animals of all ages through physical and fecal contact. Aside from CPV, there's also been reported strains of raccoon parvovirus and even blue fox parvovirus. And then CPV2 itself has diversified into smaller variants like 2A, 2B, and 2C. But for the purpose of today's presentation, I'll be talking about 2C because it has unique genetic features, it has a more widespread prevalence, and of course, it's still being studied today, so there's a lot left unknown. And if you're a visual learner, here is a visual that I created of the strain evolution timeline, and I'd like to direct you guys to today's present, where CPV2C continues to circulate globally and impacts both domestic dogs and wildlife. And here is a tree of CPV2 spread among species. So not only has the virus itself evolved, but so has the amount of species it is able to target. So today it can target over 8 species, and among these, almost half of them are endangered, and you can see those ones in blue. Okay, next thing that I want to talk about is the pathology of the virus. So essentially how the virus behaves. And you may be thinking to yourself, why do I need to know this? Like, just tell me about the solution. Well, first of all, it helps in understanding what areas and systems that the virus targets. And then in terms of that, it then reveals the crucial steps in the virus's process. And that has helped provide crucial information on how to combat the virus. So the common symptoms of CPV are lethargy, lack of appetite, severe diarrhea, nausea, and fever. And hopefully, as we talk about why or what the virus does in the body, it'll make more sense for why these symptoms occur. So it targets the 2 most fastest multiplying cells, the first of which is the lymphocytes and the epithelial cells. And then it affects 2 different systems, the lymphatic and the gastrointestinal. And I'll go into more about this in just a second. So here's a raccoon just for an example, and here is these arrows that are showing us where the virus goes once it enters the body. So first it can enter through the nose, the mouth, the ears, whatever. Then it'll enter into the lymph nodes where it'll find the lymphocytes, which are the white blood cell that are responsible for fighting off foreign bacteria. So essentially, if CPV2 can attack what is supposed to defend against it, it's almost unstoppable once it has entered. Then it rides these lymphocytes through the bloodstream, and it ends up either in the bone marrow where it finds more lymphocytes, or it ends up in the small intestine where it targets the epithelial cells, which are the lining in the small intestine. So by destroying the lining, the gut bacteria can enter into the rest of the body and cause a fatal infection. Okay. So next, once it's inside the cells, it can recognize and bind to the receptors on the host cell. And because it targets the fastest multiplying cells, it can use this lock and key mechanism and rapidly replicate within the body. Not to mention, it uses capsid protein specifically to bind to the sialic acid residue on the host cell membrane. And here's a picture of that lock and key mechanism in action. And then just to visualize the effect of it, here's a global map of canine parvovirus cases. So I used a bunch of different reports across the world of different patients that have entered clinics with symptoms of CPV. And I put that into the map just so you guys can visualize the global impact of canine parvovirus. So now I'd like to talk about the most important part, which is my proposal, antiviral peptide therapy. So in terms of application, APT can be designed very, very specific to a single virus. Not to mention it can target those viruses' proteins, which is a quality known as being virucidal. Not only that, but scientists in the past have used APT to target very specific stages in the viral process, such as viral entry, synthesis, or assembly. And considering the virus's path, which I just showed you guys, CPV's antivirals would combat viral entry, which is the most crucial part. In terms of the specifics, what we have decided is that the antiviral peptide will essentially mimic the host cell's cellular receptor and compete with the binding site. So if the CPV2 virus binds to the antiviral rather than the cell, it has effectively prevented it from entering the cell and replicating or causing that damage. For CPV2 specifically, that receptor is called the canine transferrin receptor, also known as TFR1. So this is what the APT will be developed to mimic. Not only that, but the benefit of using APTs is that cell-penetrating peptides can be added to the chain so that it can facilitate the entry of the antiviral into those specific areas, such as the bone marrow or the small intestine. And lastly, and the effectiveness. Okay, so the reason why antivirals are so effective is that, first of all, they are a biologically active molecule, which means that they can easily be broken down by an enzyme peptidase. So this makes the therapy less likely to accumulate in areas and cause toxic side effects like pituitary damage. Not only that, but APT is very small and easy to synthesize, and it can be made to be very specific, especially with the help of today's modern research. So overall, APT does pose a very promising solution to the widespread effects of CPV2. Not only that, but today the only significant therapy being used is monoclonal antibody therapy. So while antibodies, something you guys may have heard of, are much more efficient to develop, antivirals are more specific to the virus and accurate in the long run, which is especially important because our virus, CPV, evolves a lot. So antibodies depend very heavily on the geometry, and they don't work if the virus undergoes even the slightest of changes. And the potential side effects, because there's not a lot of research being done on CPV specifically, I used information from antivirals being used on HIV virus to show that the only symptoms that were reported were very mild, like headaches, rash, or mild allergic reactions. And of course, it can be potentially varied based on the factors such as gender and genetics, and in our case, species. And lastly, what this novel proposal means for the future. So a big question is resources. Unlike modern human medicine, animal medicine is simply not as high of a priority, which can make it very hard to gain financial funding, as well as resources like labs and materials. The next thing is administration. Unlike humans, where you can just call them into the hospital and administer therapy, antivirals, especially in endangered species, can be very hard because not only are the species very big, but they also live in the wild. The next is demand. I'm sure some of you haven't even heard of CPV, which just goes to show that, of course, the demand for such a solution is simply not as needed. And lastly, policy. Environmental policy can prevent us from being able to develop such a therapy and administer it to certain protected endangered species. So just to wrap up, the first one is the impact of canine parvovirus. Then we talked about the CPV pathology, then APT itself, and of course, the mechanics of the solution and the potential future obstacles. Thank you so much. And do you guys have any questions?
Thank you, Nisha. Yes, you have a question. We don't have time to answer all of those, but let's do one question here. The question is, this is a fascinating topic. If given more time, how would you further extend your research?
Yeah, so actually, I'm currently getting it published in a journal. And as part of that process, I was able to get a professor to review my work. And this professor is from Cornell University, and he works with antiviral research itself. So by building that connection, I'm looking forward to working with him and seeing more about what antivirals, like how to build antivirals, so that I can then apply that to this research and use it further. And we'll just see where it goes.
That's very exciting. That's great. Well, that's very impressive as well. So I hope, yeah, excited to see the outcome of that.
Thank you so much.